Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about the chapter 2 or part 2 of chapter 2 of your 12th class NCERT biology book, which is sexual reproduction in flowering plants. In my part 1 video I have discussed about the general structure of your male and female reproductive organs and their development. In this video we will discuss the leftover part of this chapter. So at first, the pollination. What is pollination? It is the mechanism of transfer of pollen grains which is released from anther to the stigma of the pistil, which pistil of a flower. So it can be of three types. The first one is jetenogamy, autogamy, jetenogamy and genogamy. What is autogamy? Autogamy means cell pollination, that means transfer of pollen grains from the anther to the stigma of the same flower. Then what is jetenogamy? Jetenogamy means transfer of pollen grains from anther to the stigma of another flower born on the same plant. And genogamy, transfer of pollen grains from anther to the stigma of another flower born on a different plant. So basically it is of two types actually. Pollination is of two types. One is cell pollination, which is known as autogamy. Second one is cross pollination, which is known as either jetenogamy or xenogamy. Autogamy means the transfer of pollen grains from the male reproductive part or anther to the stigma which is born on the same plant. Transfer of pollen grains from the anther to the stigma of same flower born on the same plant, same flower. But jetenogamy or first category of cross pollination, transfer of pollen grains from anther to the stigma of another flower born on the same plant. And genogamy, transfer of pollen grains from anther to stigma of a different plant, flower which is born on a different plant. So for autogamy, in such flower requires synchrony in pollen release and stigma receptivity. And also the anther and stigma should lie close to each other so that cell pollination can occur. Some plants like viola, oxalis and commelina produce two types of flowers. One is chasmogamous, that the flower which are similar flowers of other species which exposed anther and stigma, which expose the anther and stigma on maturity. But there are some flowers known as cleistogamous flower which do not open at all even after the maturity. In such flowers, the anther and stigma lie close to each other. When anther dies in flower bud, pollen grain come in contact with stigma to effect the pollination. Thus, cleistogamous flowers are invariably autogamous as there is no chance of cross pollination landing on the stigma, or no chance of cross pollen landing on the stigma. Cleistogamous flowers produce assured seed set even in absence of pollinator. Then jetenogamy, transfer of pollen grain from anther of the stigma of another flower of the same plant. Jetenogamy is functionally cross pollination involving a pollinating agent, but genetically it is similar to autogamy since the pollen grain come from the same plant. Then genogamy, transfer of pollen grain from anther to the stigma of different plant. This is only a type of pollination in which during pollination brings genetically different types of pollen grain to the stigma. Then agents of pollination. Agents of pollination, plant use two biotic and two abiotic, that is wind and water, and one biotic animals agent to achieve pollination. Majority of plants use biotic agent for pollination. Only a small proportion of plants use abiotic agent. Pollen grains coming in contact with stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen grains when compared to number of ovules available for pollination, because many of them gets gets lost during the transfer process. Pollination by wind, which is anemophily, is more common among abiotic pollinators. Wind pollination requires that pollen grains are light. The pollen grains should be light and non-sticky so that they can be transported in wind current. They often possess well exposed stamens so that pollens are easily dispersed into wind current. And large, often feathery stigma to easily trap the airborne pollen grains. Wind pollinated flowers often have single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is corn cob. The tassels you see are nothing but the stigma and style, which wave in wind to trap the pollen grains. Wind pollination is quite common in grasses or Poaceae family. Wind pollination is known as anemophily. Then after wind pollination, pollination by water, which is hydrophily, is quite rare in flowering plants and limited to about 30 genera of monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower plant groups such as algae, bryophytes, pteridophytes. So it is believed, particularly for some bryophytes and pteridophytes, that the distribution is limited of need for water for transport of male gamete and fertilization. Some examples of water pollinated plants are Vallisneria, Hydrilla, Justera. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, flowers emerge above the level of water and are pollinated by insect and wind. As most of land plants, in Vallisneria, female flowers reach the surface of water by the long stalk, and female flower or pollen grains are released onto the surface of water. They are carried passively by water current. Some of them eventually reach the female flower and stigma. In another group of water pollinated plants such as seagrass, female flower remains submerged in water and pollen grains are released inside water. Pollen grains in some such species are long, ribbon-like and carried passively inside the water. Some of them reach the stigma and achieve the pollination. In most of the water pollinated species or hydrophily, pollen grains are protected from wetting by mucilaginous covering. Then majority of flowering plants use a range of animals as pollinating agent. Bees, butterflies, beetle, wasp, ant, moth, birds are common pollinating agent. Even larger animals such as primates, lemur, arboreal, rodents and even reptiles also act as a pollinating agent. Majority of insect pollinated flowers, insect pollinated plants or insect pollination is known as entomophily, and these flowers are generally large, colorful, having fragrant and rich in nectar to attract the insect. When flowers are small, a number of flowers are clustered into inflorescence to make them conspicuous for insect. Animals are attracted to flowers by color or fragrance. The flowers pollinated by flies and beetles secrete foul odor to attract these animals. To sustain animal visits, flowers have to provide reward for the animals. nectar and pollen grains are usually floral rewards. It is a very, very important point. Nectar and pollen grains are usual floral rewards. For harvesting the rewards from the flower, animal visitor come in contact with the anther and the stigma. Then the body of the animal gets a coating of pollen grain, which are generally sticky in animal pollinated flower. When animal carrying pollen on its body come in contact with the stigma, it brings about the pollination. In some species, floral rewards are in providing safe place to lay egg. A very, very example of it is Amorphophallus. It is the tallest flower of 6 feet in height, about 6 feet. A similar relationship exists between a species of moth and plant Yucca, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of ovary, and flower in turn gets pollinated by the moth. The larva of moth come out of the leg and seed starts developing. And then outbreeding devices or contrivances for the cross pollination. Majority of flowering plants produce hermaphrodite flower or bisexual flower, and pollen grains are likely to come in contact with the stigma of same flower. Continued cell pollination results in the inbreeding depression. Flowering plants have developed many devices to discourage cell pollination and encourage cross pollination to prevent or overcome this problem, inbreeding depression. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen release before the stigma become receptive, or stigma become receptive much before the release of pollen. In some other species, anther and stigma are placed at different position so that pollen cannot come in contact with the stigma of same flower. Both these devices prevent autogamy or cell pollination. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from same flower or flower of same plant from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in pistil. Another device to prevent cell pollination is the production of unisexual flower. If both female and male flower are present in the same plant, such as castor and maize, it prevents autogamy but not jettenogamy. In several species such as papaya, male and female flowers are present on different plants, that each plant is either male or female, which is dioecious. This condition prevents autogamy and jettenogamy and only helps in xenogamy or cross pollination. So these are various contrivances or adaptation to do the cross pollination. Then after the pollination, pollen pistil interaction occur. Pollination does not guarantee the transfer of right type of pollen, that means compatible pollen of same species as the stigma. Often pollen of wrong type, either from other species or from same plant, if it is self-incompatible, also land on stigma. The pistil has the ability to recognize the pollen, whether it is a right type, that means compatible, or it is a wrong type, that is incompatible. If it is right type, the pistil accepts the pollen and promotes the post-pollination events that leads to fertilization. If the pollen is wrong type, the pistil immediately rejects the pollen by preventing the pollen germination on the stigma and pollen tube growth in the style. The ability of pistil to recognize pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and pistil. This dialogue is mediated by chemical component of the pollen interacting with those of the pistil. It is only in recent years that botanists have able to identify some pollen pistil component and interactions leading to recognition followed by acceptance or rejection. So, following the compatible pollination, if the pollen grain land is compatible type, the pollen grain germinate on the stigma to produce pollen tube through one of the germ pore. Germ pores are present on the exine. The contents of pollen grain move into that pollen tube. Pollen tube grows through the tissues of stigma and style and finally reaches the ovary. You would recall that in some plant pollen grains that are celled in two cell condition, a vegetative and generative cell. In such plant, generative cell divides and forms two male gametes during the growth of pollen tube. In plants which shed at three cell condition, pollen tube carry two male gametes from the beginning. Pollen tube after reaching the ovary enter the ovule through micropylar end, then enters through one of the synergids through filiform apparatus. Many recent studies shown that filiform apparatus present at the micropylar end guides the entry of the pollen tube. It is a very important point. All these events from pollen deposition on the stigma until pollen tube enter the ovule are together referred to as pollen pistil interaction. So it is all about pollen pistil interaction. Then about artificial hybridization. It is one of the major approaches of crop improvement program. So what are the steps of this artificial hybridization? If the female parent bear bisexual flower, removal of anther from floral bud before the anther dehisce, usually from using a pair of forceps is necessary. This step is referred to as emasculation. Emasculation is the step in which we have to remove the anthers from a flowering plant. Emasculated flowers have to be covered with a bag, generally made up of butter paper, to prevent the cross contamination of stigma with unwanted pollen. This process is known as bagging. When stigma of bagged flower attains receptivity or maturity, mature pollen grains collected from the anther of male plant of your choice are dusted on the stigma and flowers are rebagged and fruits allowed to develop. If female parent produce unisexual flowers, there is no need for emasculation and the floral bud or female floral bud are bagged before flowers open. When stigma becomes receptive, pollination is carried out using desired pollen and flower are rebagged. Then double fertilization. After the entering one synergids, the pollen tube releases two male gametes into the cytoplasm of synergids. One of the male gamete moves to the egg cell and fuses with the nucleus, thus completing syngamy. Two male gametes enter into the embryo sac through the synergids. One fuse with the egg cell in the process known as syngamy and form the diploid zygote. The other male gamete moves towards the two polar nuclei and fuse with it to form the primary endosperm nucleus or PEN, having 3N or triploid. As this involves the fusion of three haploid nuclei, it is termed as triple fusion. Since two types of fusion, syngamy and triple fusion, takes place inside the embryo sac, the phenomenon is termed as double fertilization. It is an unique event to all the flowering plants. The central cell after triple fusion becomes primary endosperm cell, develops into endosperm, while zygote develops into embryo. So the zygote, which is diploid in nature, divide and redivide to form the embryo, and the primary endosperm nucleus, which is triploid in nature, divide and redivide to form endosperm. Then after the fertilization, post fertilization events occur. The first one is the development of endosperm from the primary endosperm nucleus, that is PEN. That endosperm either completely consumed by the developing embryo, as seen in case of pea, groundnut, beans before seed maturation, or it may persist in mature seed, seen in case of castor, coconut, and used up during the seed germination. Then embryo develops at micropylar end of the embryo sac, where zygote is situated. Most zygote divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to developing embryo. Those seeds differ greatly at early stage of embryo development or embryogeny, are similar to both monocot and dicot. Figure 2.13 depicts the stages of embryogeny or embryo development of dicotyledonous embryo. The zygote gives rise to proembryo, then after proembryo the subsequent globular stage is found. Then after globular stage, heart shaped stage is found. After the globular stage, then the mature embryo of a dicot. A typical dicot embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is called epicotyle or epicotyl, which terminates with the plumule or stem tip, from where the stem will be developed. The cylindrical portion below the cotyledon is called hypocotyle, that terminates with its lower end in the radicle or root tip. The root tip is covered with root cap. Then the embryos of monocot, which is present in your figure number 2.4114, possess only one cotyledon, so it is called as monocot. In grass family, cotyledon is called scutellum. It is a very important point, that is situated towards one side laterally of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of embryonal axis above the attachment of scutellum is called epicotyle. Epicotyle has shoot apex and leaf primodia enclosed in a hollow folia structure called coleoptile. So, it is the structure of monocot and dicot embryo. Then the seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as fertilized ovule. Seeds are formed inside the fruit. Its seed typically consists of seed coat, cotyledons, and embryonal axis. The cotyledon of embryo are simple structure, generally thick and swollen due to storage of food reserve in leguminous plant. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seed have no residual endosperm, as it is completely consumed during the embryo development, seen in case of pea and groundnut. In case of pea and groundnut, no endosperms are found. Then albuminous seed retain a part of endosperm, as it is not completely used off during the embryo development. For example, in wheat, maize, barley, castor. Occasionally in some seeds such as black pepper and beet, remnant of nucellus are persistent. This residual persistent nucellus is the perisperm. It is a very important point. Integuments of ovules hardens a tough protective seed coat. The micropyle remains as a small pore in the seed coat. This facilitates the entry of oxygen, water during the seed germination. As the seed matures, its water content is reduced and seeds become relatively dry. The general metabolic activity of embryo slows down. The embryo sac enter a state of inactivity called seed dormancy. If favorable conditions are available, they germinate again. Favorable condition like adequate moisture, oxygen and suitable temperature. As ovule mature into seeds, ovary develops into fruit. The transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into wall of fruit called pericarp. The fruits may be fleshy, as seen in case of guava, orange, mango, we eat the fleshy fruit, or they may be dry, as seen in case of groundnut, mustard. Many of the fruits have evolved mechanism for dispersal of seed. In most of the plant, by the time of fruit developed from the ovary, other floral parts degenerate and fall off. However, in few species such as apple, strawberry, cashew nut, the thalamus also contribute to fruit formation. Such fruits are called false fruit. Most of the fruits, however, developed only from the ovary and called true fruits. Although in most of the species fruits are results of the fertilization, there are few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits, which are developed without fertilization. Banana is one of the example. It is very important point. Parthenocarpy can be induced through application of growth hormones, such fruits are generally seedless. Then seeds offer several advantages to angiosperm. Firstly, since reproductive process such as pollination and fertilizations are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal to new habitat and help in species to colonize in the areas in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variation. Seed is the basis of agriculture. Dehydration and dormancy of matured seeds are crucial for storage of seed and can be used as food throughout the year and also raise the crop in the next season. So then what is apomixis and polyembryony? What is apomixis and polyembryony? Although seeds in general are products of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce the seeds without fertilization is known as apomixis. Apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seed. In some species, the diploid egg is formed without reduction division and develop into embryo without fertilization. Then, what is polyembryony? Most often, as many citrus and mango varieties, some of the nucellar cell surrounding the embryo sac starts dividing and protrudes into embryo sac, develop into embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryos in a seed is referred to as polyembryony, and it is most commonly seen in case of citrus fruit. So it is all about your sexual reproduction in flowering plants. In my next class, I will discuss another chapter of your biology book of class 12th. I hope you really enjoyed this session and get benefits of it. So please like, share this video and please subscribe to my channel Sunanda's Tutorial to get more videos like this. So stay tuned and enjoy. Thank you.